new drug discovered to block multiple COVID-19 variants in lab. A new drug has been discovered capable of blocking several variants of SARS-CoV-2, the virus that causes COVID-19. This drug showed effectiveness in preventing severe symptoms and treating other respiratory coronaviruses. This paper is the first to show that activating an early immune response therapeutically with a single dose is a promising strategy for controlling the virus, said study senior author Sarah Cherry. Researchers at the University of Pennsylvania based their study on the interactions between the novel coronavirus and human lung cell lines. When the virus enters the lungs, it initially targets epithelial cells in the respiratory tract. The innate immune cells will respond after detecting the viral signature expressed by SARS-CoV-2. However, when researchers observed the interactions under the microscope, they found that the virus could hide to delay the early recognition and response of the immune system. With that critical clue, they started to search for drugs that could prime the immune response of respiratory cells to carry out a normal response. To find the drugs, researchers conducted high-throughput screening of 75 substances that target sensing pathways in lung cells. Using microscopy, they screened them for effects against viral infection, and 9 candidates came out with excellent performance. 2 of those were cyclodinucleotides, or CDNs, that could greatly suppress infection. These CDNs could do it by triggering sting, resulting in the stimulation of interferon genes. Although promising in microscopy, CDNs are not very good at making medicines. So they tested a newly developed small molecule sting agonist called diabzi. This new molecule is not yet approved by the US FDA, but is currently used in clinical trials to treat some cancer types. In their own study, Penn researchers discovered its potential to inhibit the infection induced by the original SARS-CoV-2 and its variants, including the South African variant. To prove its potential, the team used engineered mice that had been infected with the coronavirus. They administered diabzi into the mice's lungs via nasal delivery. Analysis in different biological metrics showed that treated mice had less weight loss, had significantly reduced viral loads, and had better cytokine levels compared to control mice. The findings, published in Science Immunology, are promising in preclinical trials and may unlock a new treatment for COVID-19. Beyond COVID, researchers determined that the same molecule shows inhibitory properties against the human parainfluenza virus and rhinovirus in cultured cells.